So aside from the three major vesicles of the brain, we are also seeing on this whole mount structures that are indicative of the lens forming part of the animal, the optic vesicles. Also, you can see the heart forming as one tube, then the extensions of the heart, which will actually become the Vitlin vein. So the endocardial extensions of the heart, the Vitlin vein. So these Vitlin veins connect later on to the network of blood vessels of the yolk sac. In the middle of the Vitlin veins, you can still see the opening of the foregut, known as the anterior intestinal fold, or the anterior intestinal, not fold, but portal, the anterior intestinal portal. On the sides of our spinal cord, we can see the somites, the paired block-like mesodermal cells. These somites are aggregates of mesodermal cells. Notice also that the primitive streak at this stage already grew or already regressed in a very small size. So furthermore, 33 hour stage embryo, we can see the development of blood islands situated in the area vasculosa. These blood islands are aggregates of mesoderm cells that actually will give rise to blood vessels and blood cells. Alright, let's examine the anterior view. The anterior view of the animal now shows the early developmental uh, um, stages of the five vesicle brain, though it's not very much clear, or not yet so clear at this time. We can expect to see the five vesicles in a later time, particularly in the 48 hour stage. There will be a complete impressions of the five vessels. Alright, so here we can see the divisions of the rhombencephalon, which are the met N and the myelencephalon. The non-divided midbrain, which is the mesencephalon, and the more dorsal structure of our forebrain, which is the diencephalon. Diencephalon is related with or associated with the optic vesicles. Alright. Posteriorly, we can see here that the neural folds on the lower portions of the animal are still not fused. So you expect, if you make a cross section here, neural folds are not yet fused, are not yet forming a closed tube. Now, making a sagittal cut, longitudinal cut on our 30 or through our 33-hour chicken embryo, you can see the three divisions of the developing brain, neuromeres associated with the rhombencephalon, and the heart region here. This one's a heart that is found enclosed within the pericardial cavity. Lowered or posterior to the neuromeres, you can see somites. So probably this cut was made along the somites, that we were able to see them on the longitudinal axis. So, for your convenience, we're now going to review serial sections of our 33-hour chicken embryo. For your reference, we put here the whole mount of the 33-hour embryo, and then every red line here corresponds, or every red line made perpendicular to this embryo will be represented in a bigger picture. So this red line here that's made along the forebrain, this cut here that's made along the forebrain corresponds to the prosencephalon part of the animal. Prosencephalon. This one is a more defined picture, or more defined cut, where the lateral outgrowths are impressed. So we can see the optic vesicles emerging as lateral outgrowths from the prosencephalon. Let's go downward. Here we're seeing a cut made along the mesencephalon, the midbrain of the animal, and you are able to appreciate the presence of the cavity, the not cavity but the initial part, the anterior part of the gastrointestinal tract, which is the foregut area of the animal.